Oh, you set it already up.
Yeah, so just put on a microphone and then here's the clicker for the slides. Yeah, so next slide,
Forwards,
previous
backwards.
slide, and then hold this for the pointer.
Okay.
Yeah, yeah. She also does.
Yes. I usually watch the videos later though, because I have a class at the same time.
Are you opinion in this lectures? I don't think so. Yeah. Oh no, in video lecture the class. Yes.
Oh, you've actually seen the video. Oh,
Yeah.
yes, no. Okay. I'm probably bring your voice. Chairs are cozy.
Hmm?
Comfortable chair.
I don't know about that.
That's not highly great. Okay. You want? No, no, yeah. I feel just the views. Yeah.
Whole life is
That-
Guelph
that's
motto
what-
or it's
that-
your motto?
That's what we do collectively.
Okay. Right now, in the workshop,
Yeah.
at CICR, as well, the gut for microbiome.
I know.
Now,
I know. Emma Ellen Werko is talking right now at 2 o'clock about
I don't know.
this
I
equipment.
passed by there.
Yeah.
Coincidence. And I said, where should I go? But I'm not sure I can go.
Mhm.
It's probably a little bit, you have to register.
I do not.
you
I don't know.
have to
Kathleen
pay,
Wilke.
you have to do something.
Yeah, Kathleen Wilke says she's going over there, and my colleague from Guelph,
I
Emma
was
Ellen
thinking
Werko,
about
who
you
does
when I
the.
saw this.
Yeah, I should be there too, but I also want to be here.
Like that?
Maybe I should do so-called 3D printing.
Maybe.
You from where to? The formation.
What's that?
The formation of where.
Yeah, yeah, field draw, well.
Is that it? No, I guess Samira is here.
See,
Sif wanted to come. He said he would be here.
he sent in.
Yeah, he said he would be here. I know that Thomas isn't here. Patrick left yesterday. Julia was upstairs. Hi, Sif. Oh, it's always here, isn't it? Sif, these are my students.
Oh, good. At least we've filled the room up a bit.
As Misut said, Moscow State University motto is host plus speaker plus one, you have a seminar.
That's right.
Sif is, you're not department chair anymore. He was for a long time the department chair at Applied Math in Waterloo.
They both sort of, you took them about Guelph?
Yeah.
Yeah, train, okay. Yeah. I think there are a couple more I saw out there who should be coming down, like.
I would think they should, I don't know. Does it look upstairs? Is Symmetry here today?
Symmetry, she said she was hoping to be there.
Uh-huh.
She should, if she didn't, then she said she wouldn't be there for the thing, so she was in band.
Yeah, last week she was
Last
at
week,
right, yeah. Julia was upstairs, right? Is she? Is she? Well, I don't know if she might not be interested. We don't want to force her. I don't know. It's exactly two now. Should I to Lucy?
Hi.
Can you see the slides, or should I do? I don't know the system here, or do I? Should I share it?
No, I can see the slides.
You can see the slides. Okay, good.
It's interesting because, when I was in Bangalore,
Yeah.
two of the students I was talking to, about, suggested a problem which was about the gut microbiome sort of axis. So what they did was they showed that actually administering probiotics actually helps in immunotherapy. So
Yeah.
the PD-1, PD-L1 checkpoint blockade. So we had a preliminary model, not that, though some people had published something in Math Biosciences on the gut microbiome and impact. So I think this looks like
Yeah, we looked at those things too.
Yes.
Our thing I have a master's student revisiting this. So to Lucy, who is there, she looked at that. It was a little bit inconclusive, but I think we tried to do too many things at the same time.
Yeah.
So I want Sophie to
Yeah.
sort it out and take it step by step. Right.
So Thomas also is not here, right?
No, he's flying back now.
Yeah.
Actually,
I wanted to talk to him. I sent him a thing about the Alberta math, the computational math position.
Okay. Yes.
Because I mean, I also talked to some of the computational math people. Because I wanted to know whether somebody who straddles both.
Yeah.
I mean, it would be good for them.
Yeah.
The person there told me, who's retired from computational math, that Chair's keen on the VMAX.
Okay.
I wanted to check with Thomas.
Yeah, but I'm sure Thomas will not be on that.
Yeah.
I should take the microphone. I'm sure Thomas will.
No, it's not. You might know the Stratton two areas. It'd be a very different
All right, I think that's it.
I'm Symmetry in this.
You're Symmetry today. Good.
Introduce Hermann, who you all know, and I've known for a long while, and on an interesting topic that I've also got interested in recently. So.
Yeah, thanks, Sid. Should I close the door here? So I guess everybody knows that I'm not a math oncologist. So this is a topic, I talk on a topic that I have more interest and expertise. And it's a talk where I want to lead up to that question. So I want to ask this question, I want to discuss this question. And the idea of the talk is to give a background so that we can talk. So what do I actually mean when I say gut reactor models? And this is something I'm actually qua my background, so over the last 25 years, my primary work area was bacterial biofilms, so microbial systems. And I've done other microbial things as well. And one of the things that we started about I think in 2010, working on was gut modeling. So the microbiome in the gut. When I said we, I mean a few students of mine, Arun Murthy, who did both his master's and his PhD with me. You see 2011, the master's, 2015, a PhD. Then Tulasi Chekati-san, who just defended in February. And currently I have Sophie Vadivel. They're all I don't know, interesting, is it coincidental or is it, does it come one topic? They're all in the biophysics program. I supervise in two programs, in math and in biophysics, and typically we don't notice a difference, right? What they do, but they're all, so maybe it's a topic. So Arun was from undergrad training, a chemical engineer, to Lucy, who is on here. She came to me with a master's in computer science and undergrad degrees in math and biology. And Sophie's undergrad training was in biomedical engineering. And a few collaborators that I want to mention here. Emma Ellen Werko is in the Department of Molecular and Cellular Biology. She's a gut microbiologist. And actually at the same time that I'm speaking here, she is speaking one block away at the Ontario Center for, how is it called? The OCIR, the Ontario Cancer Institute, on pretty much the same topic, right? So gut reactor equipment that she has and whether this would be useful for the research. So I should be there in Emma's talk, but I also should be here. Martin Kalmakoff and Steve Brooks were two government researchers that sucked me into this. Martin, I guess I can say he's retired. He was at Agriculture Canada, and as far as I know, he's doing commercial real estate now. He's an anaerobic microbiologist who took early retirement from Agriculture Canada. Steve Brooks, Health Canada by training, a biochemist. He was for a while the director of the Bureau of Nutrition of Health Canada. He didn't like that job, so he got himself demoted to become a scientist again. And so where my involvement with gut reactors started had nothing to do with gut diseases. It was the healthy gut from a viewpoint of functional foods and nutraceuticals. So the question is, do probiotics help? Do prebiotics help? Stuff like this. And Martin and Steve thought, if they confer benefit to the host, then the first thing where you should notice would be in the gut, right? Which makes sense. So this is where we get in, and we got initially lots of money for this from the networks of centers of excellence, and General Mills was the industrial partner for that. I said initially because we don't have this money anymore. Then the Ontario Ministry of Agriculture, Food and Rural Affairs put money in that. This was, again, I said, more than 10 years ago, and functional food and nutraceuticals were a big topic, and lots of money was available for this. And of course, since then, it's funded by NSERC. So here are our papers on gut. There are not too many. There are six papers that I could find there that are really on gut modeling. The first are from essentially Arun's PhD, and then the second half is from Toulouse's. And as you see, we sucked Arun into this in them too. And so this is what I talk about. Here is the gastrointestinal tract from this, I'm sure, very scientific webpage, Bugs in Your Guts. So we see there's what happens here, right? Just look at the pH. We start neutral, then we get to something very acidic. And what we are talking about is really the end of the GI tract, the colon. Commonly, one distinguishes between three parts. That's really a difficult cursor to move. The ascending colon, the transverse colon, and the descending colon. Sometimes they call this the distal colon at the end, and these two are together called the proximal colon. And you see the pH is different in the stomach. So stomach is a very harsh environment for bacteria, right? It's a very different bacteria that you find here than in the gut. So that's what we're looking here at the green part. So the colon, which is the primary organ of the lower gastrointestinal tract. The function is, well, it's the final stage of digestion, to remove water, to process indigestible material such as fibers, and fermentation of carbohydrates. There are lots of bacteria there, and a healthy individuum has hundreds, thousands of different species. Most of them have never been cultivated. And I think we all have a different population composition, but there are some that probably many of us have. And important for some of the things we've been doing is that the lumen and the mucus have different populations. So what are the lumen and the mucus? That's here. This is now another thing that's a more scientific source here to Lucy through this. So here's again the colon, and here Lucy made a cross section through the colon, and then we see a lumen, and the lumen is surrounded by the mucus. The mucus covers the intestinal epithelium. It's a physical barrier. It separates the microbes from the host. It offers attachment sites for the microbes in the lumen, and it supplies carbon to the gut microbiota. So it applies food to the gut microbiota. And there are two layers: the inner mucus layer that is free of bacteria, and the outer layer is colonized by gut bacteria. And when I said before, here, distinct luminal and mucosal populations, this means they can just shift. So it's not exactly the same, right? But there might be a different strength relative to each other. Okay, so that's the system that we're looking at. So why to study the colon? The first 3 points is what I said already, the role of the colon. Then there are certain important fermentation products in there, and this will come back in the model. We have short-chain fatty acids that are absorbed mostly by the host. We have specifically these three: acetate, propionate, and butyrate that have an effect on the host, right? So short-chain fatty acids and gut health have become quite important. And of course, there's the relations to gut health and gut disease, right? The gastrointestinal diseases, of course, have to do with the colon, but not only. We have non-gastrointestinal diseases like T2 diabetes, type 2 diabetes. Bone quality is related to gut health, celiac disease, certain cardiometabolic diseases, non-alcoholic fatty liver disease. They're all related to the microbial population. The gut-brain axis is an important thing. I think there's sort of connections that Alzheimer's disease is related to the microbial ecology in the gut, right? I don't know too much about it. And the reason why I'm here and talking about this is this one: connections of gut health to cancer. And this would be primarily colorectal, what we would think of, but it could be also others, right? So this doesn't show as well as it should. This is sort of a hallmark of cancer wheel that we probably have seen before. And this is from this paper, Fulbright et al. From 2017. So here are a couple of the hallmarks of cancer. So this is not all of them, but some that are related to the intestinal microbiome, right? And here there are a couple of players are individually pointed out. So one that will come back is Bacteroides theta iota omicron. So it will show up later in the talk. I also want to point out the butyrate, one of the short-chain fatty acids that I mentioned before, that also the model that I talk about will track. So there's quite some of the hallmarks of cancer are affected positively or negatively by what's going on in the gut here. And this is my angle. This is where I'm coming from. So how can we connect? Can we connect a model of the intestinal microbiota to a cancer model, right? So the microbiome and hallmarks of cancer. Of course, the first one is Helicobacter pylori infections. They are not of relevance to us. They live in the stomach, right? They live under very different conditions than but I think these are the first. That was the first bacterial species that was related to cancer, right? But we're not talking about that one. And the second bullet point is what I showed you in the wheel before, that the resident microbes likely influence tumorigenesis or by modulating hallmarks of cancers. And if we understand this, then one can have an additional target for treatment or prevention, right? This is true probably for many microbiomes, not just the gut microbiome, that the relationship between the bacterial population and the cancers, right? But this is a very microbially a very rich environment, the gut. And then another point, and this is, I think, something that makes everything difficult, is that the cancer microenvironment can enhance procarcinogenic activities of the microbiota. So it's not just the microbiota affects cancer processes and tumor growth, but the tumor, what the tumor does, the microenvironment of the tumor also affects the microbiota. Hermann.
Associated with ulcer, stomach ulcer.
Yeah, with some cancers, I'm not sure, but I think yeah. But
Stomach-
most of us probably have it. So just that
no,
you
the
have.
question is, stomach ulcer, is this because of H. pylori, right?
I do not know what the could be. I don't know what the exact.
Yes, I mean, it's implicated, but it's not.
I don't know if it's the cause or if you have stomach cancer, and otherwise
The
this means.
question would be, if someone has stomach ulcer, do they have to worry about cancer?
If somebody has stomach cancer, do they have to worry about cancer?
Stomach ulcer.
Yeah, ulcers. Yeah,
ULCR.
yeah, ulcer. I don't know. I'm a gut guy. I'm not a stomach guy.
Okay.
Can I just, Hermann, just
Yeah.
reiterate, it's actually in the last generation of the hallmark of cancer, the microbiome itself is.
It was,
hallmark
yes.
of cancer.
Yes,
So
yes, I saw that. But I sort of don't. I think it's sort of a silly thing, right? How can something that we all have a microbiome, right? I hope we do. Well, I guess if we wouldn't have one, we wouldn't be here. But how can this be a hallmark of cancer in the same way as the uncontrolled cell growth and stuff like this? So I'm a little bit. I don't quite like that. But yes, I'm aware of that, and I might come back to this in another slide. But you're right. It is now.
Yeah, I also don't never understand like this hallmark of cancer. They've been increasing and it's like some of the time it's just cancer adjusts something that is in the system more than it be a fundamental.
Yeah. So I'm sort of, but again, I don't know oncology and math oncology, but yeah, thanks for mentioning that. I sort of tried to not mention it. I try to avoid that.
Yeah.
So how is the colon studied? It's very difficult to assess and control experimentally, right? You cannot just get in there, and at least in the living system. Fecal sampling is one way, but people see what's going on in the colon, but there you only see the distal, so just essentially what is eliminated, right? You only see it only represents the distal lumen, right? So you don't see what's in the mucus and stuff, right? So what people are doing is they have this kind of sorry, I guess I'm going in the wrong direction here. This kind of experimental systems. This is a schematic of SHIME, the simulator of human intestinal microbial ecosystem, which is, I think it's Jan van Impe, somebody in Belgium. I think it's Jan van Impe who builds and distributes that. And so it's a series of, well, as you see, flasks. They're all treated as shaked flasks, right? There is one, the first, oops. No, I'm going in the wrong direction here, sorry. Begins with the stomach, so a very acidic environment. This is where the food and additional acids are put in. Oh, I'm sorry. Then we have something that, one that mimics the small intestine, and then we have three that mimics the colon. The ascending, the transversal, and the descending one. And then we have an outflow here. So this is a schematic of one of several groups here. This is, oh man, sorry. I guess I will not figure out how to use that. This is the RoboGut, which is from Emma Ellen Werko's lab in Guelph. This is how a photograph of it. Emma does more gut disease, so it's not all about gut health, what she does, but also gut disease. And so that's just how it looks. So it looks, if you want, sort of similar. There are flasks and like in the schematic, right? So that's a reactor system. And when I say gut model, or you might have seen on the slide, I say gut and then in brackets, reactor models, because I think what we model is actually not the gut, but a gut reactor, which is what lots of biochemical engineers or microbiologists used to study. So if you had a question?
No.
No. Okay. All right. So this is sort of the setting that we have in mind when we do a model. And here's an in silico version of it. So this was an in vitro setup. This is an in silico model. So we have here a dual compartment system. It's like chemostat setup, right? We have one reactor, one compartment, that's the lumen. This is where we have on inflow, and in the basic model, we would only put in the fiber, which is what feeds the bacteria. We have an outflow, which all substances that we have in the lumen, they flow out. And then we have what chemostat people call sometimes a lateral diffusion compartment, right? This is a compartment where we have exchange of material between this compartment and that, but there's no external inflow and no outflow other than here the absorption to the host, which is a simple sink term in the equation. So as far as the exchange goes, and I still haven't told you what the components are of the reactor. So there are bacteria, there are short-chain fatty acids, there are a few other components. And the reactions we assume here to be the same in the lumen and in the mucus compartment. Here's the reaction pathway for our gut model. And this is based on the anaerobic digestion model #1 of the International Water Association, which is sort of a standard model in anaerobic wastewater treatment. So it's an adaptation of a wastewater treatment plant to describe the gut. Just some parts of it, it's not the full thing. Certain things that we have in the wastewater, we wouldn't consider here. So we have fiber as our source of everything that's degraded here in hydrolysis, enzymatic hydrolysis into sugars, converted into lactate. Then the utilization of lactate creates these short-chain fatty acids. We also get water, carbon dioxide, hydrogen. Then there are species that converts hydrogen into acetate or hydrogen into methane, or uses it for the production of this. Martin Kalmakov tells me that a third of people don't have methanogen producers. So not everybody has methanogen producers, says Martin. And yes, so this is the pathway. So this is essentially the diagram of what's in our model. Again, I said it, it's based on the IWA anaerobic digestion model number 1. The model comes from Rafael Munoz Tamayos. I think that's a odd story, which I won't tell you now. And then Arun extended it. The key idea here is that I told you there are thousands of bacteria, right? You cannot track all of them. There's no specifically if we don't have them, are not able to cultivate them. So the idea of this model and all of the ADM1, anaerobic digestion model number 1, and other models that the International Water Association has put together, activated sludge model, the river quality model number 1. When I was on a biofilm task group of the IWA, we didn't want to put a model together, a quasi-standard model. But so the idea is that the bacteria, they are grouped by functionality, not by kinship. So we don't look at each species individually. We look at species as a group of species. These are bacteria that degrade sugar. These are the species that produce propionate and stuff like that, right? So that's sort of the idea behind this grouping, functionality grouping. In our model, the only carbohydrate, as I say, the only carbohydrate source is fiber. And that's hydrolyzed in sugar, as I've shown you in the pathway. Short chain fatty acids that we track, butyrate, propionate, acetate. Butyrate, I pointed out before, right? That was in this hallmarks of cancer wheel from that in core. And other products we have in the model are water, carbon dioxide, hydrogen, methane. So how does the model look like? It's an ordinary differential, a system of ordinary differential equations in the simplest case. Key to this is the so-called Peterson matrix that has n rows and m columns. The components of the system are organized per column. So we have 14 here. These are our biomass species, our short chain fatty acids, the carbohydrates, etc. And then we have each process refers to a row, and here are 9 that are included. So it's in our case, it's a 9 times m matrix. The process reaction rates, they take the components and map them into process rates. So very, very simple. The matrix entries are yield and conversion constants. The reaction terms then, and this is not the full model, the reaction terms then are given by this here. Now you might ask, so if it's called the Peterson matrix, why is it called S and why is it transpose? And I'm glad you asked. I don't have an answer. I assume that in chemistry, they call the corresponding matrix the stoichiometric matrix. So S transpose, right? Is this true? I don't know. Could it be true? Maybe. I don't know. I just made this up, right? So I have no intelligence about the origin of this notation, but it's typically written as the transpose of a matrix, this reaction terms, which is odd to us, right? So for each of lumen and mucus, we have 1. I told you before, we consider in the model the same species in both things, the same reaction are going on. So the matrix S, we have 1 matrix S for the lumen, 1 for the mucus, where they might be different, could be parameters, right? But this is all the same. C, here are our components, right? So this is a system of 14 ordinary differential equations. We have 2 components, so it's a system of 28 ODEs that we're looking at. So this is the Peterson matrix. It doesn't fit on 1 page, so I had to split it up. There are 9 processes here. The first 5 I have also here for the first 9 components. Here are the last 5 components. And you see here if you write this as a block matrix, if you like it, write it as a block matrix, we have here on the right side, we have here 2 diagonal matrices, and here we have a sparse matrix if you want. I'm not sure that sparsity plays a big role. It's a small matrix, so I don't think we get anything out of using sparse matrix properties here. And there's an empty block there. And so this is the model, how it looks like, where for both together, we have here the Peterson matrix. Here is the process rates for the lumen, for the mucus. These are the minus D. Here is the washout rate, the dilution rate for the lumen, and the other, the DLM and DML, and with the head and without the head. This is the exchange between the compartment, the lumen and the mucus. And this is the input vector. I told you there's nothing input into the mucus that's not quite right, because the mucus produces mucins, so that they're autonomously, so we don't add it externally, but it's produced in the mucin. So in the compartment diagram, I should have made sort of a circly arrow to the compartment. And this UL, that's the fiber that we add. These matrices D are non-negative and diagonal. So that's essentially our model. And again, it's a 28 differential. Jonah. Because the mucus and the lumen have different volume, and we're talking in concentration, so we have to account for that, right? So there's a constant. So DLM and DHLM, there's a factor difference, which is the ratio of the volume of those two compartments, right? And I'm not sure why there's a T. So that's a typo. Right, so that's essentially the model. So just to give you a feeling for the model, we have 28 ODEs. There are about, I don't know how many parameters, 40, 50, 60 parameters in it. So lots of stuff, lots of uncertainty in those models. I want to talk very quickly about one application. Oh, sorry, I lost my microphone here. Thanks, Marcus. So, very simple study, response to antibiotics. We expose the gut micropopulation to antibiotics, not to kill the bacteria. We wanted the opposite. So we are concerned, how long does it take for the population to recover from the antibiotics, right? And then, of course, we can ask the question, then of course, we can ask the question, if we add probiotics, so bacteria on inflow, how would this affect the recovery? If we add prebiotics, prebiotics are non-live food ingredients that confer a health benefit to the host. So we don't have much choice for this. The prebiotics in this model setting, it could be either fiber, eating more fiber, or short-chain fatty acids, right? So these are the changes that we have. Probiotics could be any of those 4 functional groups of bacteria without changing the model, right? And yeah.
Do probiotics survive the pH of the stomach?
That's actually a very good question. Do probiotics survive the pH of the stomach? I guess they have to be encapsulated accordingly.
Okay, so they're protected.
Yes, so you don't when you go to wherever you go, the pharmacy or the health food store, and buy probiotics, and don't just take a spoon of bacteria that you, so, right? They're encapsulated in something, and then the question is, do they get through this? And actually, independent of what I want to talk about here, one thing how I got into this was that Lisbeth Brustrup Hansen, who was at that point a microbiologist at Dalhousie in the food science department at Dalhousie, she's now in Denmark. She wanted to use EPS, extracellular polymeric substances produced by biofilms. So she wanted to look into, can we create those, encapsulate the probiotics so that these biofilm produced EPS protect the probiotics on the way through the stomach, right? So yes, so let's assume this here, when we add bacteria, this is, they survive the stomach. They're somehow protected to get through them. Again, it's a very important point. I guess the same thing you can ask about prebiotics, right? If you take a spoon of butyrate, what goes on in the stomach, right? What happens to your butyrate in the right? You can, or does it even make it, or does it? I don't know. What it does to you. Yeah, that's probably one thing. Yes, and yeah, But I won't touch this because I think we try to investigate too many things at the same time there. So prebiotics, probiotics before, during, or after the antibiotics. So it became very messy. And I want now Sophie to take a step back and look at this more systematically and just look at one thing. All right. So this is from one of Toulouse's papers. This is for something that we call a strong antibiotic. Strong or weak is just one parameter. I should say we're looking here at bacteriostatic antibiotics. So antibiotics that don't kill bacteria, but that prevent them from growing. It may or may not look the same if we take bactericidal antibiotics that kill bacteria. We haven't looked at it for this. We have looked at bactericidal antibiotics for mostly when we do antibiotic stuff for biofilms, we look at bactericidal, and here we do bacteriostatic for whatever reason. Or for reasons that I don't want to go in now. Julia.
I asked something about the model before. Is there any cooperation between the bacteria? So like, are they eating what the waste of other bacteria is? And I guess it's somewhere in this matrix. Right?
Yes, it's somewhere in the matrix.
So there is
right?
cooperation.
Yes, well, cooperation, there's one, well, I guess I go back to here, right? So there's not between all of them, right? So the sugar degraders convert the fiber into sugar and degrade the sugar, and then the lactate degraders do all of these things. And then here we have a hydrogenic bacteria that produce acetate as well, or
So
methane.
the proliferation depends on
Yeah.
how much, like the waste, what they produce is the waste. Of their
Some,
own
yes, so
bacteria.
there's some interaction. It's in, as you said, it's in the matrix. Where is it? The matrix is not full, so not everybody interacts with everybody. So, but if you look here at the rates, so X1 here, bacterial species 1 will be multiplied here only with X1, X2 we have, so mostly they all do their own thing, right? But via the short chain fatty acids or this, yes, and here you see how these processes, glucose utilization, lactate utilization, they're all interconnected, right? So it's not that you can completely decouple it and look, instead of having the system, you look at for ODS.
There is no negative effect, so no way, no product is toxic. Any of them.
No, it might be inhibitory, so there might be inhibition, but
not-
yeah, but not. So they are the natural decay terms, but they are all first order, right? X1 decays proportional to X1. So the natural decay terms don't depend, but they're inhibition effects, right? Via pH, for example. Okay, so this is where I was. So in the simulation, and we can only study this really by simulation, there's very little you can do analytically. We run the simulation to steady state, then we hit it with antibiotics. So the antibiotics was added here to the model that I said before, is another concentration. It's just on or off, right? And then here the sugar degrader in this strong antibiotic, the sugar degrader goes down to what appears 0, right? But very small. The other ones come back very quickly. The sugar degrader also comes back quickly. This is in the lumen. In the mucus, that looks pretty much the same at different levels, right? But the substrates, the short chain fatty acids, methane, and so on, sugar, they look, there we get different pictures qualitatively. I don't want to get into the details of that. If we're looking at this is the more interesting thing, weaker antibiotics, right? Yes, the sugar degraders are hit by the antibiotics. They don't go very far down, but then they come back again, right? The other biomass species, you don't really see much. So the conclusion from this was that according to this model at least, the response to antibiotics is really, it's that on the top of the chain, right? The sugar degrader, they sit on the top and they control everything, how the system recovers. So that's what we took from that. And that's what we want, right? We don't want the antibiotics that we take because we have an ear infection to kill our gut bacteria, right? Next thing. That's here continuously over a small time window, right? So when here we go up to equilibrate and then we hit it with antibiotics, then we see where the sugar degraders come down, and then when the antibiotics are turned off, then they come back again. And this time is what we measure, right? And this is here in the order of, what is it, a week or two. And this might agree with our experience. Once we took antibiotics for whatever, it might take us a while until everything goes back. But that it has to go back, or antibiotics would be a really bad thing, right? So having seen that it's really, at least for the antibiotics that we look at, those that do not completely eradicate the microbiome, that is the sugar degraders, then what we did is we're just looking at the top chain, conversion of fiber into sugar, and then the consumption of the sugar. So we just focus on the top level group, the dietary fiber, the sugar degrader, and the mucins. I didn't tell you about the mucins yet. I said it once, I didn't call them mucins, but I mentioned it somewhere. Mucins is produced in the mucus, and that's carbohydrate resource for the mucus population. So it's essentially the dietary fiber would correspond to what we have in the lumen. And you may remember that I told you fiber is not exchanged between the lumen and the mucus, because in the mucus we have mucins that are produced there. So we looked at this. This was one paper. This is another paper that I'm talking about now, and I will tell you in a minute. So instead of having just one sugar degrader that we had before, we're looking now at two. We're looking at competition between a generalist that would consume, which we had in the previous model, that would consume the fiber and the mucus. We add now to the model. So we first chop the model off, just look at the top part. And now we say, no, the sugar degrader, we add another species to it, right? This means we have another column, another row in our Peterson matrix. So it's a different model. And these examples for generalists versus specialists, so for us, it doesn't really matter. It's parameter values. But two examples for a generalist and a specialist are Bacteroides theta iota omicron, which you might recall. I pointed it out before in the wheel chart. It was in the inner core of the wheel chart. This is a generalist. And then there's Akkermannsia muciniphilia as a specialist. And there's a paper from last year. And thanks to Lucy for sharing this. The title is called Akkermannsia muciniphilia, Friend or Foe in Colorectal Cancer. So the studies that looked at the role of Akkermannsia muciniphilia, and this is now I guess in mouse studies, they found, some say it helps promote cancer, others say it helps to slow it down or suppress it. And this article tries to reconcile those results. So this is where the title comes from. So this first reduced and then extended model has now 11 ODEs and 39 parameters. So still lots of stuff in there. There's nothing we can do analytically, right? We can do computer simulations, what we want for this paper in JTB. So we try to do the simulation stuff. We try to do it properly. Lucy ran 6.5 million simulations of the model for one question, right? So, but that's cheap, right? We can create data as we want for that. And so these are the highlights that come out of this study. The competition for glycans can explain mucus thinning during dietary fiber deprivation. So we don't get enough, the host doesn't get enough dietary fiber, the mucus might be thinning, right? And this is something that we saw in this competition model that we have not seen before without the competition. So we don't know that, is it? But could explain, right? Then if you have dietary fiber deprivation, the abundance of the mucin specialist, Akkermansia muciniphilia, the one that we don't quite know yet if it's good or bad for us, right? But which most of us have. The abundance increases, and then mucin specialist benefits from sugars, freed by glycan generalist through cross feeding. And the last one is sort of a technical thing, the computational approach that we took, that a random forest classifier machine learning technique can help really to provide insight into the long-term behavior of a model. That's sort of a more, there's nothing to do with the biology behind it. Yes, Julia.
Can I ask something about this last point about the random forest? So the idea is that what's the input? Like you give the parameters and it tells spit you out.
The input, well,
Steady state?
the idea, yeah, the thing, the starting point for the whole thing was that when we do ODE analysis, dynamical system analysis, we look at types of equilibria, right?
Specified types.
Classifying types of equilibrium, whereas if we do sensitivity analysis in ODE models, we always look at steady state values, not at the types. So the sensitivity analysis that we now, for the last 5 years, have been doing routinely to overcome our algebraic limitations to deal with the classification, what the traditional dynamical system is, said, well, why don't we try classifier, machine learning method to classify if this helps? Now, it's a big model. And to loosely classify, there are, I guess, if you want, in terms of coexistence, absence or presence, or in the lumen and the mucus, there are only, well, either you don't have bacteria in either, or you have both. You have bacteria group in both. You don't have that, you have it in one.
So you
And-
just give it
but-
this studies the last time point of your simulation.
No, you
steady
give,
state?
you
you
make sure that,
keep-
it's a steady state,
Okay.
right? So this,
Yeah, yeah, yeah.
this six
But
and a,
then it's
you have to,
kind
you
of
have to check this, yeah. And then you, the idea was, can we then sort of, these inequalities that we get in the ODE models, and these parameters satisfy these inequalities, then you're in. But that's too big, what you get here, right? You get this random forest classifier, put everything into boxes, essentially, into intervals. And, but you get just too many. We have 39 parameters here, right? But it did help to design simulation experiments. It didn't give an easy to use answer for this. And this is something I would like to investigate, just starting from a simple chemostat model where we know everything about it, whether this classification methods, random forest classifier, can help, or when do we have to lose confidence in what they tell us about classification, right? By building up complexity, but that's, I guess, something for another student to do. And as I said, there were in this study for this year, there were 6.5 million simulations to steady state for 6.5 million different parameter values went into that. All right, so this was that. Now I'm coming back to that, what I showed you before. The butcher rate affects here, this hallmark of cancer. The Bacteroides theta iota omicron affects here. These are things that we had directly in our model. The Akkermansia, we don't have in there, but we have the other paper by, what was it? Gubernatorova from last year that looked at relationship of Akkermansia to muciniphilia to cancer. And then we have here other things, lots of other things that we have not in the model, but well, I guess the bifidobacters, this would be sugar degraders, I think, right? They would be now a top group. So I guess they're also there. And this is a popular probiotic, the bifidobacters, right? So some of those things, some of the bacteria, they are in those groups that we're looking at, and then there might be also other short chain fatty acids if they produce any. So I don't know about the green guy here, who they are. or this blue guy, what it does. So how does it affect those things? So what would we have to do? We have already some? It's a matter of looking into it. I told you I'm not an expert in those things, right? So this would be one question, is how to look into when I come to my question now. Are this type of models useful in Math Onco, or could they be useful in Math Onco? One could first look what of the model that we have already shows up in this simple, and of course we should probably not overemphasize that, right? That is just this just is essentially a summary of what Fulbright et al. Understood in 2017. I don't know if they would draw it again in the same way now in 2024. But this is sort of a potential starting point. This is how I think about this, right? So the model itself, how can we extend it for Math Onco? And I'm just speculating here. If we need new microbial species with new functionalities, we can put this into our model framework, right? Again, we add a row and a column in the Peterson matrix. The same thing is if we need chemical species, if we need special chemical species, we might need to also have to add additional bacteria. Some of the chemical species might be produced by the tumor that might affect gut microbiota. So this is the other way back. More challenging is if we have to put in host physiology, which we don't have in the model. Very simple. We have from the lumen extraction term of short chain fatty acids. And if we have to combine this with immune system aspects, then we would have three things: Math Onco, immune systems, modeling, which I also don't know, and the gut stuff well, Toulouse knows it better than I do, but that we have experience in. So in any case, we cannot expect that this will permit insightful analysis, because already here, just in the gut model, we have our limitations, right? And I assume it's the same with many cancer models, right? At least from the seminars I've seen in the last weeks, that there's just too much going on. They usually have the virtual patients with the parameters and this and this and that. So, and this is you do because you can't write the analysis then, right? And this needs to be explored in painstaking numerical experiments. And this is so this would be sort of the methodology that would need to be used for this, I think, right? So this is my outlook. Here we have a gut model that affects the hallmarks of cancer, that affects tumor growth. This is what our wheel told us for Fulbright et al. We also have that the tumor might affect the gut model, so it might go back. Does it go through the hallmarks of cancer? Likely, I don't know. Or is instead the tumor affect just an arrow directly to the gut model? I do here now what Thomas did in his first class, in the Math Onco class. He writes down the most generic tumor model, the logistic equation, and then whoever, I don't know who was in that class, right? And then he says, okay, now he went through the hallmark of cancer around the wheel and said, this hallmark of cancer, which of those two parameters, growth rate and carrying capacity, does it affect? Right, and that's so essentially this means that these parameters, the two parameters that the logistic equation have, they're either affected by a hallmark of cancer or not. So each hallmark of cancer might affect both or one. So we can write this down. Now, what we have here, so the starting point here is that the gut microbiome might affect the hallmarks of cancer. So this means now that the hallmarks of cancer, the H, might be a function of M. And so here's our most simple way of writing a tumor model that takes the microbiota into account. Logistic equation where R and K depend on all the stuff that we have in the microbiota model, or on some of the stuff, right? And in which sense? this is the thing one has to look at. What's in the other direction? So this would mean, does the tumor microenvironment affect the process rates so the stoichiometric coefficients in the Peterson matrix, or the processes, our functions rho, that we multiply against the stoichiometric? And if so, how, right? So this is sort of where my thinking goes for that. And my question is here, is there place, do you think that there's place for this type of models to be used in math oncology? And if so, is anybody interested in exploring that further? And this is, with this question, I would normally say, and now I'm open to questions. Now I'm open to answers. Chile.
So I see that there are like, this is more on the bioinformatics side. So like, I'm not an expert in this, but I know there are many controversies going on about all of these bioinformatics analysis of gamma and so on, whether it's really connected or not to cancer. You know, because I guess people have done like bioinformatics studies to see whether there are connections on it. It seems like it's very, people disagree a lot. So I guess, are people doing some more, because I can imagine that like so we know that tumor metabolism is very important in tumor progression, and the metabolites of the tumor are alongside those chains, so they can ejaculate and so on. So I guess maybe are people doing like in vitro experiments of this, or like
That's a good.
we have an understanding of how?
You mean in vitro experiments of this in the connection to cancer?
Yeah, I mean,
I
imagine if you could culture this bacteria with the tumor cells, there would be.
Yes, and my understanding is there's some going on the bioinformatics side. I cannot speak about this at all because I'm coming here from the wastewater side. We're looking at function and not at kinship. Right? The genetics will give you the kinship. So who is there? Who's related to whom? But we are for this model setup. We're more interested in what do they do. And I don't know the genetic side and I would, so this is a very ecological approach, right? This type of modeling.
Yeah. But I was wondering in general, like what you envision is like, because we said it could be a tumor that is in the colon itself, and then you have direct. Cross-talking, or is it like, how does this affect our own physiology?
Yes, so and I think metabolomic studies that look into this, who is there and what is there, certainly one has to look into that, right? But I'm not quite sure whether the data would be good enough to parameterize models like this or to inform model like this. So what's missing? Is it important? The thing is, my understanding, my very limited understanding of this whole bioinformatics side is it primarily tells you what's there and not what they do. Right? It's more descriptive. It's more descriptive. It's not really function and dynamics. And so this is what we're after here, function and dynamics.
In a sense, there has been some work done, and mainly coupled with immunotherapies. So if you look at immune checkpoint inhibitors like PD1, PD-L1, then it's been shown that some patients respond very well to the study and others don't. And there's been evidence now showing the gut microbiome diversity between individuals has a lot to do with that. So we've looked at a much simpler model, and let's say looking at how probiotics can enhance or prebiotics. And from the computer simulations, it looks like certain concentration of probiotics above a certain range actually enhance the immunotherapy. And, and they compare it with the fecal matter transport. So probiotics seem to give you a much better boost to the immunotherapy. Whereas prebiotics from our circulation seem not to help very much.
I guess it always then depends, as I said before, prebiotics could mean just a fiber, so or it could mean, well, what are we now sort of interfering with a short chain fatty acids and other chemical species in the directly. So I think there are so many wheels that you can turn.
So probiotics, I think, suggestion, but from our things, could be a natural enhancement for immunotherapy.
Yeah, yes, probiotics, I think why they are one of the reasons why there's a little bit of skepsis why the people don't agree on it, whether they work or not. It seems to depend very much on the individual, how the individuals respond. I have seen it work with a cat, but I've also seen it not work. Right? The probiotics don't help with anything. And this is, again, when I say a cat, I'm not claiming all cats, but Catalyst, the cat, responded to it.
Stay on the immunotherapies, like what's the mechanism behind, like why are probiotics helping? Is like
Yeah.
they boost the immune system or they, what, I guess my question is like mechanistically,
Yeah.
why are these affecting?
Because, well, I don't know, my understanding is sort of a bit, the PD-1 receptors on the killer cells. The cancer cells have the PD-L1 ligand, and they sort of inactivate the whole thing.
Yeah.
Somehow the probiotics deal with the PD-1 receptors in some way, you get some, I don't know the biology, yeah.
It might be just an ecological thing, right? You have a population, then you throw a whole bunch of individuals in there. And by whole bunch, I mean a whole bunch, so not just two in a population of 2 million. And then things might start to shift, right? And that shifts everything. Then it shifts.
No, yeah, that's
So-
why like I'm interested in terms of how this like
Yeah.
feedback happens. It's like, are they changing the absorption of are they changing like, yeah, how
Maybe
what
that-
happens in
yeah,
our gut?
What I'm
like,
saying is maybe
influence?
they're just changing the ecology, right?
Yeah,
I mean, somehow they sort of seem to mitigate the immune cell, how the cancer cells sort of inactivate the immune cell.
Yeah,
So somehow,
directly, there's some direct.
yeah.
So are you changing the metabolism of the tumor itself?
That's a good question. Yeah.
That's why I just like, yeah, it's very interesting, but I just like.
But it sort of certainly fits in with what you said about personalized therapy, because it depends on the gut microbiome of each
yeah.
individual.
Yeah.
Because certain bacteria seem to.
And also that if they do studies that are so inconclusive, right? That seems to suggest the study objects must play a big role, I think, right? So either it's a completely arbitrary process, or does the subject, the study subjects play a bigger role, right? Than Western medicine asks, right? For the approval of things, it has to help pretty much everybody, right? Or at least not harm anybody. In a study. So that's sort of our standard.
There's a paper by Chen, 2022, which was in mathematics bioscience. That was about the first OD type model, so about what the effect of gut microbiome on therapeutic effect. And so we took that model and then added 2 more ODs to simulate probiotic.
Yeah.
So it's just purely a simulation. But we found that it sort of confirmed some of the observations, but we don't have a mechanistic explanation exactly what's happening.
Yeah. But I guess it's like it's very many things. I can imagine the gut influence is basically all of our physiology. So it's kind of, but I guess it's not like I guess it's not that it's a microbiome of the tumor itself, but it's more like
Yes.
the effect of what happens in our gut is in some way influencing all of our physiology as well as the physiology. Because that was something we did all of these hallmarks of cancer. I'm not sure whether they refer to like the tumor itself as its own microbiome or it's like generally our microbiome.
Yeah, that's how I understood it, and that's why I thought that I don't think it. I don't know why it is there. It doesn't seem to be of the same quality. Which one?
There are 2 processes which are working in the reverse direction because gut reactive modeling is saying that want the bacteria to live, and cancer model, we want the cancer cells to be killed.
Yes, but I'm not suggesting here to use the gut model framework to model tumors, but to connect the gut model to a tumor model, right? To our logistic equation, for example. So that's what I wanted. And
Is there any understanding of how changes in the gut microbiome influence nutrients in our blood, for example, when you like?
I don't know. There could probably there's, but I don't know. So again, these models, we call them gut reactor models because we really don't look at the host much other than the absorption of the short chain fatty acids out of the mucus, right? So that's the other question. What are these reactor models, right? What do they actually tell us about a living? But I think we have this everywhere with all in vitro systems, right? That are used to study anything.
So I'm just curious why we started thinking about it when I was listening in the Institute of Science.
Mhm.
I just gave a couple of lectures and then I just floated the idea about looking at the gut microbiome and a couple of the undergraduates they went and researched that and came up with that first model that became the book. And then they sort of extended it around simulations. But I think there must be more work in that direction.
Yeah, I don't know. So Thomas tells me, and I don't know anything about math onco, right? And I don't. The literature is vast. I don't even personally want to start looking into it, right? Because if you start, I think it sounds like a hole that you never get out. So I think people who know this literature much more, right? Whether they know what's there.
That's a much complex,
Yeah.
mechanical problem. So the
Yeah.
But as you say, the oncology.
That's just very difficult. So and I don't wanna start on this alone. If I don't know where it leads me, and if I don't have anybody to guide me where to go there. So if anybody wants to look into this, I would be very interested.
Started with the Aspergillus bacteria. It is actually I suffered from this a couple of years ago. So it is a bacteria that is like a quiescent, and it is in our stomach.
Yeah.
As the food is digested with the acid
Yeah.
of our stomach. So when we got this is awakened this bacteria, it is already in our stomach. And then what happened, like the acid of our food like destroy the walls of our stomach.
Yeah. So
It becomes a wound, and if we do not treat it,
yeah, that's what Idema asked,
it
right?
becomes ulcer,
Information.
Yeah.
like free
Yeah.
like an ulcer, and then it couldn't be
Yeah.
turned into the cancer.
Yeah.
So, but it is specifically like the stomach cancer.
It's a stomach. Yes, that's why I just mentioned it because I think historically it was the first one that was connected to cancer, but it's stomach. I don't care about this. I shouldn't say that.
Sometimes it is curable with the medicine, and sometimes
Yeah, yeah.
But you do not want to consider just a stomach cancer or something like this. You want to understand that how this is connected with the tumor growth or the cancer growth, all over the body.
Yeah. So yes, that
This like this way can be connected because whatever we take the treatment medicines that goes through our stomach.
Yeah.
If it doesn't work properly, that might be we can connect with, like, we cannot cure the tumors, we cannot treat the tumors because of this bad
Yeah.
work.
Yeah.
Cannot treat.
Yeah.
Like this.
I don't know, but again, I don't Stomach is a very harsh environment, right? So I don't want to go anywhere near there.
Some of, we do have multiple treatments. So some of
And that's the other question. How do things like this, right? That we might have cancer in the stomach or other gastric cancers, how do they connect to the microbiome then in the intestinal microbiome? Right? There are lots of things, of course, one can look at, but I think just because there are so many one can do this doesn't mean one shouldn't do any. And then one might as well start there where one has in my opinion, most advanced starting conditions, right? Which probably is that. We are not the only ones who do gut modeling. There are a couple of other groups. There's a group in Edinburgh and Kettle. How is it called? Biostatistics Scotland, I think is the group called. And there are a few others, but not many, right?
The immune checkpoint inhibitors, particularly those targeting PD-1, PD-L1 interaction, they have marked a significant step forward in treating solid tumors in patients. But as we said, it's a sort of varied response patient to patient.
Yeah.
And the link seems to be somehow the difference in the gut
Yeah.
microbiome.
So would you say, when I sort of carry on my question here, would you say that if one wants to look into that, one also has to look into the whole immune response things, and there's no point in doing it without that?
Well, that's my point of view at the moment.
Okay.
Maybe it's which aspects you're looking at. If you're looking at the PD-1 type of drugs, yes. That's immune therapy. But I guess maybe if you look at, I mean, it always depends on the starting question that you want to answer. And it's like, you want to ask about, I don't know, if there is any crosstalk between the metabolism of the tumor and like whatever happens in the gut. That would not require immune system, I guess.
Yeah.
It would be required to capture more the metabolism of the tumor. Some simple way, but again, I don't know.
All right. So now I put the idea out there.
I mean, I think during the first workshop, there was like Thomas that did wrote their proposal on the microbiome or something. So they might know a little bit Thomas Hillen.
Yeah.
I knew no idea.
Yeah, he could.
About the
He's
literature.
on his way back now
Okay.
from Alberta, so he couldn't be here. But I hope to talk to him one of the next few weeks.
Yes. He's taught it.
No.
I sort of how the tulips get their stripes. Apparently there's some mutation of the tulips which have the stripes. So it wasn't oncology, but it looked like a viral or something.
Okay, I don't wanna keep you longer. Does anybody else have other thoughts, other insight to share, other questions, other complaints?
I was just wondering whether there is, talking about Thomas as well, whether there is any interaction between the microbiome and the biotherapy, for example, as another kind of treatment that doesn't require immune system, I guess.
Yeah.
Yeah. But I think it's a sort of area of increasing interest, but not so much well established. Certainly having emerging evidence.
All right. What's now? Tea?
What is this?
The workshop, yeah.
Is it that problem solving thing?
Yes, yeah, exactly. So